Good morning. Welcome back to Biochemistry 601. This is lecture number 14. Technically it's lecture number 14, but we're still on lecture number 13, but I will catch up. Now, many of the things that I talked about last lecture are written up in this chapter, Enzyme Catalysis. It's chapter 8. You can find it on Canvas, and it's free. So if you're interested in more details, please take a look at this. I also should let you know that I'm not alone in the room. Mr. Bear is still here. And today, Mr. Bear is going to learn about cofactors. But the last thing we were talking about in lecture was the orientation being very important. And what you see with enzymes, especially when they have two substrates, they get them aligned. It's perfect. It's orientation. It's orientation. And a lot of reactions that go on in biochemistry are SN2 type reactions. You know this from organic chemistry. S is for substitution, N is for nucleophilic, and 2 is for bimolecular. And it results in inversion of configuration, and the trajectory doesn't deviate by more than about 10 degrees. So it's 180 degrees. It's single inline displacement reactions. And that's shown schematically right here. So you can see inversion of configuration around that atom right there. So I'm going to show you an example. And this is the case of N-acetylgalactosamine kinase. And what does this enzyme do? It puts a phosphate group right there. It uses ATP to do it. In the human enzyme, the structure that I'm going to show you, we still don't know what's the physiological role of the human enzyme. It's thought that it may be important in a salvage pathway, but we still don't know. But what you need to see here is that you're going from that hydroxyl group to a phosphoryl group right there. All right, so the structure of N-acetylgalactosamine kinase was solved. This is a ribbon representation of the human enzyme. You can see the nucleotide. This is the adenine ring right here, and the sugar binds right there. Now this is really neat because the first structure of this enzyme was solved in the presence of the sugar and a non-hydrolyzable ATP analog called AMP-PMP. And that was first worked out by Ralph Yap, who was a professor now retired at Washington State. Ivan's going to tell you more about non-hydrolyzable analogs of ATP when he talks about ATP-dependent enzymes. So the purple is what you see when the structure was solved in the presence of AMP-PMP, which cannot be hydrolyzed, and the sugar. And this is the OH group that's going to get that phosphate group. And then the structure was solved in the presence of ATP, and the sugar as well. But when the structure was solved, when you looked at the active site, the substrates have actually turned over and got trapped in the active site. So then what you see is the ADP, the phosphate group, which would have been over here, is transferred to that hydroxyl group. And there you can see the inversion of configuration going on. So in summary, for this lecture 13, we talked about acid-base catalysis and concerted acid-base catalysis. We talked about covalent catalysis, metal ion catalysis, electrostatic catalysis, and this proximity and orientation effects of getting things together so that the reaction occurs. These are some of the concepts that enzymes use to carry out reaction. Now, these are the classifications of enzymes. This is just for your education. You don't need to memorize this, just keep these notes. But what should you focus on for this particular lecture? You need to be able to push arrows correctly, and we all make mistakes pushing arrows, so work at that. You must know the difference between nucleophiles and electrophiles and what's going on there. Understand general acid-base catalysis. Understand covalent catalysis. What are the roles of metals in catalysis? What does this mean, catalysis by approximation? And this is an important one. Understand how the pKa value can change depending upon where that residue is in the protein, its local environment. That's important. And we didn't talk it in this lecture, but we did talk about transition state analogs in the kinetics lectures. So you need to understand, why are transition state analogs such good inhibitors? What's going on? All right, so now we're going to turn our attention to lecture number 14, but this is chapter number 9. So if you're interested in the material that I'm covering here, then please take a look at this chapter. Now, cofactors and coenzymes are something you could take a whole course on, a whole semester. Some of the chemistry that goes on is just stunning. I just sit there going, that's unbelievable. All right, so there's 800 or more than 800 different types of reactions that go on. And a lot of these reactions that enzyme catalyze can use acid-base chemistry, covalent catalysis. But there are a lot of reactions that cannot do that, right? And then you think about it, there are no amino acid side chains that can function as electrophiles. And these amino acids can't accept electrons, so there's a lot of reactions. You know, in glycolysis, you're moving electrons around. You need cofactors. You need cofactors. And so this next lecture, lecture number 14, we're going to discuss these 3 cofactors. And then in the next lecture, we're going to talk about some other cofactors. But these, we already know a lot about nicotinamide adenine dinucleotide. We've talked a lot about that already in this class. So these are good. These are good for hydride transfer. You see them a lot in metabolic pathways where you've got to be moving electrons around, hydride transfer. But they're limited. Now the flavin adenine dinucleotides, those are amazing, some of the chemistry that goes on. And we still don't understand it all. Even though it's in a textbook, don't assume we understand it all because we don't. And then there's thymine pyrophosphate, TPP. Now there's not that many enzymes that use it, but the enzymes that use it, the reactions are absolutely stunning. So what are cofactors? They're non-protein molecules that function as essential parts of some enzymes. Not all of them, but some enzymes, right? They can be metal ions. We talked about metal catalysis, but they can be organic molecules. Now, the cofactors, coenzymes, it's semantics. I'm not careful about it. Just know that they're non-protein molecules. They're non-proteins. And they're also referred to as coenzymes. They're organic molecules. And they're often derived from vitamins, which we'll talk about. And what happens is there's special reagents, right? They can do reactions that the regular amino acid side chains can't do. Amino acid side chains are good nucleophiles, and they're good acid bases, depending on which ones you're looking at. But they cannot accept electrons, and they cannot carry groups around for the most part. And so that's what these cofactors are necessary for. So the common coenzymes, cofactors, and I said you can take a whole course about this, but these are the ones that you would typically see. NAD, this NAD or NADP, it's oxidation and reduction reactions going on. The flavins, they're also oxidation reductions. You're either gaining electrons or losing electrons. Leo the lion says girl, loss of electrons is oxidation, gain of electrons is reduction. This is where you're moving aldehyde groups around. TPP, stunning chemistry. And the pyridoxal 5' phosphates, we'll talk about that next lecture. They're involved in amino group transfer. Folate, probably my favorite cofactor, folate. That's the one that I love to talk about. Coenzyme A, it's okay. It doesn't have the kind of chemistry that folate has or pyridoxal 5' phosphate, but it's still an important coenzyme. And then there's biotin. If you see something and it says the enzyme is biotin dependent, you better believe that it's involved in a carboxylation reaction. It's carrying carboxyl groups. And then there's S-adenosylmethionine, that's involved in methylations. Now, an aside. And I made some comments about Linus Pauling, and I said I would get to it. So Linus Pauling was the one who started this hybridization stuff, which explained what we see in the laboratory in terms of carbonyl carbons that are sp2 hybridized, that kind of information. He was also so brilliant that he figured out long before protein structures had been solved that proteins were going to curl up into helices. He understood the potential of the hydrogen bond. He won 2 Nobel Prizes, but he also almost had Nobel Prize disease where he thought he knew everything. And the reason why we have this vitamin supplement right now going on, we can thank because of Linus Pauling. So on October 10, 2011, researchers from the University of Minnesota found that women who took supplemental multivitamins died at rates higher than those who didn't. 2 days later, researchers from the Cleveland Clinic found that men who took vitamin E had an increased risk of prostate cancer. It's been a tough week for vitamins. But these findings weren't new. 7 previous studies had already shown that vitamins increase the risk of cancer and heart disease and shorten lives. And yet, how many people take supplements? It's amazing. We have all these stores, these supplement stores, and it's not regulated by the FDA. And they're probably doing more damage. And some people say, well, they can't do any harm. Yeah, they can do harm. They can do harm. And believe me, I used to take supplements until I read this book, Do You Believe in Magic? And I have to tell you a story. So I was reading it when we were visiting our Canadian relatives, and they're kind of in all this health food stuff and all that kind of stuff. And I'm kind of trying to stay, I didn't want them to see that I was reading. And I got to the chapter about vitamins, and I said, that's it, Ivan, I'm not taking any more vitamins. And he goes, they can't hurt, they can't hurt. And then he read that chapter, and when we got home, he threw the vitamins away. So this is another book I would strongly recommend reading, because if you have a good diet, you don't need supplements. And the supplements, it's a reductionist kind of view. It's like, well, vitamin E, so we'll give you more vitamin E. Actually, you don't want to do that. So it's interesting. But we can thank Linus Pauling, who was so brilliant, for now leading the way for all these vitamin stores. All right, I'm off my soapbox. So now we're going to talk about the pyridine nucleotides, and you've heard this in this class now. They're either NADH or NADPH, and they look like this. This is NADP, all right, because the phosphate group is hanging off that adenine ribose. In NAD, it wouldn't be there. But then this is the business end, the nicotinamide ring, and it's that carbon there is C4 carbon, and when that thing is sp3, it is a pro-chiral center. Now when the hydride leaves, remember you have the Re and Psi face that we talked about. In this case, the nicotinamide ring, if we're looking at it, it's the Psi face because we're going in the counterclockwise direction. Now, the adenine nucleotide part is very important for binding, but it's this part right here that's important, the business end. That's where the hydride gets transferred. And where does this come from? This is niacin. This is a vitamin that you take. It's vitamin B3. All right? It's also known as niacin. Deficiency can cause pellagra. Now, this is an interesting one. This niacin was first purified by Conrad Elvehine in 1938. He was a professor in this department. He was born in McFarland. He served as chair of the biochemistry department. He was the grad school dean. He eventually became the university president. And there was this museum down the street named after him, the Elvehine Museum. Well, in 2005, the Chasins gave a whole lot of money and they changed it to the Chasin Museum. But in my mind, it's still the Elvehine Museum. So let's just take a look at the NAD. All right, one thing is that in solution, it kind of curls up on each other, but when it's actually bound to enzymes, it's in an extended conformation. The sugars are both D-ribose. Now this is important. In the cell, the concentration, the ratio of NAD to NADH is 0.1 to 600, depending on who's doing the measurement, so they're hard to make. But the point is, in the cell, you typically have more NAD plus than NADH. And if you look at catabolic pathways, when you're breaking down food stuff and you're grabbing electrons, if you're oxidizing things, something has to get reduced. So if you look at the reactions in catabolic pathways, if they have hydride transfer, they tend to use NAD. Now some enzymes can use NAD or NADP, but in catabolic pathways, you tend to see it being NAD. Now, in the cell, the NADP plus versus NADPH ratio is near 0.01 to 0.005, which means you have more NADPH, which is a reducing agent. And so if you look at anabolic pathways where you're building up molecules, you're doing reduction reactions, it makes sense that in those pathways, they tend to use NADPH. Now, how do we know that its stereochemistry goes on? How do we know that these enzymes are either A site specific or B site specific? How did this come about? How do we know this? And we don't talk very much about experiments in this class, but let's talk about this one because this is classic. Right, so what these researchers did is they took alcohol dehydrogenase and NAD, which has NAD, and then they took this one didural ethanol here. So you have deuterium here instead of hydrogen. And they carried out the reaction and pushed the reaction in that direction. So what is that? This is getting oxidized and the NAD is getting reduced. Right? This went from sp2 hybridization to sp3. This now is a pro-chiral carbon, but in this case it's actually chiral. Right? So then they take this and they made this, and then they go back and they take the monodural NADH and they mix it with acetaldehyde, that's acetaldehyde, and they push the reaction. Now, if this thing wasn't stereospecific, you would have expected the deuterium to end up in some of the molecules, but not in all of them. Right? So we're going from this oxidized to reduced. And if it wasn't stereospecific, you would have expected some of the molecules of the ethanol to contain hydrogen there, right, instead of the deuterium, and that's not what they saw. So it's a classic experiment that showed that these enzymes are stereospecific. And there's the A side versus B side specific enzymes, and you have seen this slide before. In the A side specific enzymes, such as malate dehydrogenase, right, the nicotinamide ring is in the anti-conformation. It's not hanging over the ribose. You're looking at the re face, so it's clockwise here. In the B side specific enzymes, such as glyceraldehyde 3-phosphate dehydrogenase, what you see is the nicotinamide ring is rotated about 180 degrees about that glycosidic linkage right here, and now you're looking at the side face. So these are A side specific enzymes and those are B side, and that's how they have evolved. So the B side specific enzymes have the nicotinamide ring in the syn conformation hanging over that ribose. Some of the general properties of NAD and NADH, and we've already talked about this when we talked about assays. So it's not really that important to go through this again, but just to remember that NADH or NADPH has a good absorbance at around 340 nanometers, and that is often used in these coupled assays. And here's another example of these coupled assays. In this case, we're looking at UDP galactose 4-polymerase. What does it do? It changes the configuration around the C4 carbon right there. It's an enzyme involved in the lower pathway. But you can't measure that. You can't assay for that. So what you do is you take this, once it's made UDP glucose, you use UDP glucose dehydrogenase, which oxidizes that carbon right there. You have a COO group, and if that got oxidized, something had to get reduced, and the NAD goes to NADH, and then you can follow it through the absorption. So these are reactions catalyzed by NAD or NADP containing enzymes. This is probably the most typical, where you're going from an OH to a C double bond O. But these things can go back and forth, depending upon the physiological state, depending on how you're carrying the assay. This is another reaction that you'll see, NAD-dependent enzymes or NADP. These reactions forming a double bond. I'll show you that in a second. And then, for example, here. But this is typical what you're going to see happen for a reaction of alcohol dehydrogenase. In the active site, there'll be some base, aspartate, a glutamate, histidine, something that's going to pull that proton off here, right? And those electrons are going to get fed in here. And then this hydride, it's a hydride transfer, gets transferred over to here. And then you can reverse the reaction. Now I thought I'd give you an example of one that's important. It's called enoacyl carrier protein reductase from Mycobacterium tuberculosis. And that's the causative agent of tuberculosis. It's been estimated that 1/4 of the world's population is infected with tuberculosis, but they have a latent form of tuberculosis where they don't even know that they have it. They don't have symptoms, they're carrying it. Now, we can treat tuberculosis now, but back in the 50s it wasn't great. And when my grandmother came to visit my family, and this was before I was born, but my brother and sister were quite young, she came to visit, she was coughing up a storm, and they had her checked out, and she had tuberculosis. And she ended up in a sanatorium. They used to put these people away in upstate New York. Well, there's treatments now, and you can be cured. The problem is these treatments are arduous. They're not fun to do. But this is a drug that's used, isoniazid. Right? So this enzyme is absolutely required for the biosynthesis of glycolic acids, which this bacterium needs in the cell walls. And if you can stop it, the bacteria can't survive. So isoniazid, first described in 1912, is the first antibiotic that they typically use, isoniazid. Now you look at that, well, you know, that looks like kind of a nicotinamide ring, but there's some really interesting things over here. This looks like it could be quite reactive. And what does this enzyme do? It takes a double bond and makes a single bond out of it. So this is going to a reduction, which means that NADH gets oxidized to NAD. Now the structure of the eno-ACP reductase is shown on this slide. It is a subunit, one subunit right here. It has a Rossmann fold. It functions as a tetramer. It has a Rossmann fold. The NAD is stretched across the C-terminal part of that beta sheet. It belongs to the short-chain dehydrogenase reductase superfamily, so you know there's a characteristic signature sequence for these. This structure was solved, and all of this was worked on by John Blanchard, who is a professor at Albert Einstein, another one who was trained under Mo Cleland. And John used to say to me that, quite frankly, he would sooner have surgery in a field in New Jersey than in a hospital because of all the bacteria running around. Right, so how does isoniazid work? It forms a stable adduct with the nicotinamide ring. And once it does that, you can't have hydride transfer, so the reaction can't occur. So what is it about this reaction? How does it work? So here you have the tyrosine and a lysine, and that tyrosine is the one that's conserved in the short-chain dehydrogenase reductase superfamily. It's the pro-S, which is what these family members do. The hydride is transferred over to here. Those electrons move into here. Those pop up here, so now we have an oxygen sitting there with a minus charge, which is stabilized by its interaction with that lysine 165. And then the proton off the tyrosine 158 is removed over to here. Those come back down. You can follow the chemistry here, and you end up seeing the reduction going on. So those are NAD or NADP, and they're beautiful cofactors for hydride transfer. But what happens in reactions when you have more than one, maybe you want one electron transferred at a time? So hydride transfer, you're moving 2 electrons at a time. What happens if you have a reaction where you actually have to move electrons one at a time? So the stoichiometry of most enzyme catalyzed processes, redox processes, indicate that 2 electrons are transferred, 2. But sometimes there's one electron step, right? And sometimes it's 2, like the hydride. When it's 2 one-electron steps, they result in free radical intermediates. Now, these are some of the other ones that you see that can also transfer 1 electron at a time. Vitamin C, the thing that Linus Pauling pushed so much, which does not treat colds. Coenzyme Q, you actually should know about that one. That's involved, it's a component of the electron transport chain here. Then there's vitamin K and vitamin E. But we're going to talk about the flavins. They're interesting and they're more common. So the flavin proteins contain either flavin adenine dinucleotide, FAD, or flavin mononucleotide, FMN. Where did the flavin word come from? It comes from the Latin word, which means yellow. And when you isolate these proteins, they tend to have a yellow tint to them. Riboflavin, also known as vitamin B2, was isolated in 1933 from egg whites. Now, this is interesting. In most flavoproteins, the cofactor is covalent or bound tightly, not covalent, but bound very tightly. So with NAD-dependent enzymes, depending on which one you're looking at, but most of them, that cofactor goes in, then you have the substrate coming in, then the substrate will go out, the cofactor, maybe it's been reduced or oxidized, goes out. So they're like co-substrates. Now, occasionally there's some enzymes that hold the NAD very tightly, but most of the time, the NAD or NADP goes out, in and out. But the flavins are bound tightly, and sometimes covalently. And this allows the protein to modulate the redox potential of these cofactors. But what this also means is that once the reaction has occurred and the electrons are in that flavin, something has to come along and get them out to start the cycle again, because the flavin that's been reduced does not leave the enzyme. And that's important. That is an important fact. So, generally speaking, NAD or NADP is a coenzyme that you'll see involving carbonyl compounds, where FAD and FMN are coenzymes at other types of oxidations. Now, the term FAD is typically referred to as a dinucleotide, flavin adenine dinucleotide. It's a misnomer because the D-ribitol, which I'll show you, is not attached to the flavin ring by a glycosidic linkage. It's really a mononucleotide. So here's the structure of FAD and FMN. This is the business end of the molecule. That's the flavin part, right there. And I always mispronounce that name, so I'm not even going to try this year. It's isoallox, whatever. I have such trouble with that, so I'm going to call it the flavin ring. So what is it? You see FAD and FMN, there are the flavin rings right here. And this is the ribitol, right? So that's not a glycosidic linkage. And then you have a glycosidic linkage right here, the adenine part here. This part right here is riboflavin, and that's what you get as your vitamin, and then there are enzymes in your body that finish making it off here. So in the case of FMN, there's an enzyme that will put the phosphate group there. The flavins, in terms of their spectra, what you see is FMN and FAD have a number of absorbent maxima here, and in particular there's one at 450 here. But when this actually gets reduced, all right, dihydro FMN and dihydro FAD, that's now reduced. And when it gets reduced, you can see the absorbent maxima goes down. So when flavin-containing proteins are isolated in their oxidized state, right, they're typically yellow in color. But once they get reduced here, so they go from FAD to FADH2, that color tends to disappear, and that's because you've wrecked up the conjugation here. So what's the difference between FAD and FADH2? Well, in this case, we have a hydrogen sitting here, we have a hydrogen sitting here, these double bonds are no longer here, and that one's there. So you've broken the conjugation, and you reduce this thing by 2 electrons. That's what you've done. Now, in addition, what's interesting is that when it's oxidized, the flavins tend to be flat, but when they get reduced, they start to take on kind of a butterfly conformation. And when you see that buckle, it occurs between the axis right here, so they kind of buckle out a little bit. So, this is the oxidized form of the flavin. This is when you have 1 electron and 1 proton that's been added, so this is a 1 electron step going on. You have the flavin semiquinone, and then you get the 2nd electron and you have the reduced flavin. So it moves 2 electrons, just like NAD does, but not as a hydride, And so this is the oxidized form, the semiquinone, and the reduced form. And this is the biggest difference, because some reactions, sometimes, you have to move 1 electron at a time. You can't do that with NAD. You can't do that with NAD. That's important too, because the flavin is able to carry out 1 electron transfer reactions where NAD is restricted to 2 electron hydride transfers. Now, one of the things that's interesting is once that thing is reduced, it doesn't leave the protein. So there's got to be some kind of electron transport protein or something else that's going to react to get the electrons out of here so you can oxidize the flavin again and the reaction can occur again. Now, what we see with flavin-dependent enzymes is that they have either 1 or 2 conformations. They either look like this or like this. Where NAD, it's always extended. But in this case, they'll have either this kind of conformation or this kind of conformation. And this is interesting because it's the local environment as well as these conformations that dictate the redox potential of the flavin rings. There are 4 families. They're listed right here. I used to show structures of them. I don't think that's actually important. I think what's important is you realize that these families, there's 4 that exist so far that we understand, and they will bind the flavin either in this conformation or that conformation, depending on which family member you're talking about. These are flavin-dependent enzymes, the reaction types that you tend to see. So here's one, acyl coenzyme A dehydrogenase. And that's the first step in fatty acid oxidation, and I'm going to talk about that. And there's succinate dehydrogenase. You should know that succinate dehydrogenase is called complex 2. It works both in the citric acid cycle and the electron transport chain. And what does it do? It builds a double bond there, just like acyl coenzyme A dehydrogenase. They both use this. Now there's these enzymes called diamino acid oxidase, where you're actually oxidizing, but then molecular oxygen is being reduced to hydrogen peroxide. And that's the way it gets the electrons out, where these enzymes actually, once they've been reduced, have to work with an electron transfer protein to get the electrons out. In these cases, once the reaction occurred, oxygen comes in and it gets reduced to hydrogen peroxide, and now the flavin is oxidized again, and that also occurs here. So, some flavin enzymes function via hydride transfer, just like NAD or NADP. Others go through a carbanion mechanism. Some go through a radical mechanism. There's a lot of them out there that we still don't understand how they work. And sometimes, there's actually molecular oxygen that will bind to these rings. So it's not only moving electrons, sometimes you'll get oxygen or something else binding here, and where they bind depends about the enzyme. So some will bind at the N5 position here, and some will attack the 4A position there. So not only are they moving electrons around, sometimes they're actually part of the reaction in terms of getting hydrogen peroxide or something down there as well. So I thought I'd give you an example of one that's medically relevant, cholesterol oxidase. Cholesterol oxidase. So what does this do? Well, this enzyme catalyzes the oxidation of beta-hydroxysteroids and the isomerization of the intermediates. Pretty interesting enzymes. The structures have been done to very high resolution by Alice Beerling, who is a friend of mine and a professor in Australia. This enzyme is found in a wide range of bacteria, and it's a flavin-dependent enzyme. It's flavin dependent. So this is really neat because this is the way they use this cholesterol oxidase to measure serum cholesterol levels. So what does it do? So cholesterol oxidase produces hydrogen peroxide. So here's the reaction it catalyzes. It's first going to oxidize this carbon right here, so we go from a COH to a C double bond O, that's an oxidation. And then the enzyme catalyzes this isomerization right here. But when it does this part of the reaction, then molecular oxygen comes in and reduces reduces water to give you hydrogen peroxide. You know, you got hydrogen peroxide here. All right, so what you can do is take the hydrogen peroxide, and then you can use this enzyme, a peroxidase, that couples the reduction of hydrogen peroxide to the coupling of phenol and this 4-amino antipyrine, which gives this rose-colored compound right here. So you take the peroxidase, it takes these two compounds together, and it couples it with the reduction of hydrogen peroxide, couples the reduction of hydrogen peroxide to water here, and then you get this pink color. So you can use this as an assay to measure total serum cholesterol levels. Now let's talk about another one, beta oxidation of long-chain fatty acids. This is my favorite cycle. You have this long-chain fatty acid, that R can be 12, 13, 14 carbons, whatever. But what you're going to do is basically run the cycle around so that you reduce the long-chain fatty acid by 2 carbon units in the form of acetyl-CoA, which then can get fed into other pathways. The first step is probably the hardest in this pathway because you have to put a double bond here, and that's not so easy to do. Right? Once you've done that, that's fatty acyl-CoA dehydrogenase. Once you've done that, you add a water molecule across the double bond. That's not too hard to do. Then there's another dehydrogenase which uses NAD. This one is FAD dependent because that's a hard step to do. This one is NAD dependent. It basically is oxidizing that carbon, the C beta carbon. The OH goes to C double bond O. There's a thiolase that comes in here, and you reduce the long chain fat by 2 carbon units, and then you feed it in here again. Now the fatty acyl coenzyme A dehydrogenase, once this has been oxidized, it's been reduced, and then it has to interact with electron transfer protein to get those electrons out so that the cycle can go around again. Medium chain acyl-CoA dehydrogenase, the structure is shown on this slide. It functions as a tetramer. There's looking at just one subunit right here. This structure was solved by Jeong Kim, who is retired now, but was at the Medical College in Wisconsin, in Milwaukee. Wonderful person. And this is an interesting one because mutations in the human gene encoding the medium chain acyl-CoA dehydrogenase are relatively common, mutations in them. But there's one point mutation where A985 goes to a G, leads to a substitution of a lysine residue with a glutamate at position 304. So you end up having a change in charge of that position. Now this isn't in the active site. It just makes the protein unstable, so you don't have a functional protein. And the defects in this dehydrogenase lead to the disease state, which is referred to as medium chain acyl coenzyme A dehydrogenase deficiency. It approximately occurs in 1 out of 10,000 live births, which is pretty amazing. Now, if it's tested for and you know it's there, you just make sure that there's never periods of long time where you're fasting. Because when you fast, right, that's when your fatty acids start to get broken down. But you don't need them to break down if you're not under fasting conditions. Now, those persons with it can survive if they're very careful, but they end up having seizures, brain damage, cardiac arrest during periods of fasting or illness, especially if it hasn't been diagnosed yet. But it is tested for. But some suggest that about 1 out of every 100 sudden death syndromes may result because of this NCADD. Interesting. All right, so this is a close-up view of the active site, the structure that Jung Kim solved. Here's this glutamate, 376 right there. There's a backbone amide group there that's pointing towards, the hydrogen would be pointing towards that oxygen of the CoA part here. It uses CoA bound. All right, and then there's a hydrogen bond from the flavin, no less, forming a hydrogen bond there. And when the reaction occurs, there's development of a negative charge on that oxygen, and that helps to stabilize that transition state. It also, if you look at this, it lowers the pKa of that alpha proton. So that's also important because you got to pull that off to get this reaction to occur. So here is the proposed reaction mechanism for this enzyme, and as I said the last time, every time you see a reaction mechanism, take it with a grain of salt, because there are some people who say, no, it doesn't work that way. And when you see things written up in biochemistry textbooks, just realize that it is still a work in progress. We still don't understand a whole lot of things. But what they think happens is this glutamate pulls the pro-R hydrogen off here. Those electrons get fed into here. That goes up to here. And now you have a negative charge on that oxygen. And that's why those backbone amide group and then the hydroxyl group from the flavin is helping to stabilize that negative charge here. And then it's thought that the pro-R hydrogen, and in this case, it's hydride transfer. It's thought, but other people will say, no, it's not. But in this particular example I've drawn, they think the pro-R hydride is transferred over to here, and you end up with that double bond. And then this enzyme has to interact with electron transfer protein to get it to reset to over here. So the nomenclature. Some flavin-dependent enzymes are referred to as dehydrogenases, like fatty acyl coenzyme A dehydrogenase, or succinate dehydrogenase. Others are oxidases, and others are called monooxygenases or dioxygenases. And these names distinguish the manner in which the electrons are transferred out of the reduced flavin rings. So the dehydrogenases employ electron transfer proteins to reoxidize those cofactors. That's what they do. But oxidases, like cholesterol oxidase, uses oxygen to regenerate the oxidized cofactor with the concomitant production of hydrogen peroxide. And then if you see monooxygenase, it tells you that one of the oxygens, right, it employs oxygen, but one of those oxygens is ending up in the substrate from oxygen. One of them is, and then in the dioxygenase, both oxygens from O2 are incorporated into the substrate. So for the remaining minutes today, we're going to turn our attention to thymine pyrophosphate, TPP. Out of all the cofactors that I'm talking about, this is the only one I've never worked with so far. And I used to not know much about cofactors, but until I turned my research attention about 15 years ago looking at enzymes that are involved in unusual sugar biosynthesis, I had to learn a lot about cofactors. And there was one enzyme that actually used TPP, but we could never get it to crystallize. But this cofactor is stunning. It's not that common, but it's amazing the chemistry that goes on. And yet again, we still don't understand it all. So thymine was the first of the vitamins to be identified, hence it's called vitamin B1, vitamin B1. If you don't have thymine, you can end up with this beriberi, a disease state. But that's not going to happen in most situations where you have decent food. The coenzyme form is thymine pyrophosphate. So this is the vitamin, and when you take it, then there's an enzyme that's going to put these phosphate groups there. It serves as a cofactor in those enzymes that catalyze bond cleavages between carbonyl carbons and adjacent reactive groups. That's what you need to think about. So this is thymine. That's what you get in your diet. This is thymine pyrophosphate that is used as the cofactor here. And it always comes with a magnesium ion, and that's because you've got to stay or shield those negative charges of those phosphoryl groups right here. This is the business end of the molecule. That's what's doing the reaction, and it's absolutely amazing what happens. Now you think about this. All right, a carbon-hydrogen bond, the pKa normally, the pKa of that would be maybe 50, right? Because this is not going to want to lose a proton. But notice that you have this nitrogen sitting there that's positively charged, and that's going to do something that's important. The thiazolium ring here, that's what's going to happen. That's the business end. And here's what's amazing, is that the pKa of this is about 12.7. Now a normal carbon-hydrogen bond is 50. How is it 12? It's because you have this positive nitrogen sitting there. And then you have a strong nucleophile. It's called a carbanion or an ylid. An ylid. What's an ylid? An ylid is any neutral, it's neutral, dipolar molecule, it's dipolar, that has a positive charge on one atom and a negative charge on the other. It's amazing. So these, once you have this form of it, you have a powerful nucleophile. So the TPP-dependent enzymes, there can broadly be separated into two categories: those that function as decarboxylases and those that function as transferases, where you're taking a group. They display ping-pong reaction mechanisms. Ping-pong. That means something goes in, substrate goes in, something happens, something goes out, another substrate comes in. Remember ping-pong. You should go back and remember, how do you distinguish between ping-pong reactions? The first half of the reaction involves the breaking of a carbon-carbon or a carbon-hydrogen bond adjacent to the carbonyl group, and some also break other bonds, but what they all have in common is they bind their cofactors in a similar manner. So NAD tends to bind in a similar manner. The flavins, on the other hand, they can bind in an extended or a more bent way. These tend to bind the cofactors in a similar manner. Acetyl-CoA, that's not true of that one, but these do. So for the longest time, people couldn't, let me go back to this slide here. People thought that the active site, there had to be some kind of active site base or something that was going to pull that proton off here to give you that carbanion. For the longest time, that's what people thought. But then the structures got solved of some TPP-dependent enzymes. And what they realized, right, is what happens is when it binds in this orientation. They think there's tautomerization going on, and what happens, there's a conserved glutamate that pulls that proton off here. These electrons get fed into here. This pulls off the proton here, and now you have the ylid. So when it binds to the enzyme, it binds in a conformation that allows the chemistry to go on, so you don't need an active site base to pull that proton off here. The cofactor is doing it for you. So pyruvate decarboxylase. Let's talk about this one, because this one's important. In yeast, pyruvate decarboxylase, as the name implies, it's getting the carboxylate group off in the form of CO2, and then it's going to reduce this to ethanol, and we all know what this is used for, to make beer, for example. So this is an important reaction. So yeast, you have pyruvate decarboxylase, and then the alcohol dehydrogenase that converts pyruvate to ethanol. There you go. Now, pyruvate is an alpha-keto acid. What does that mean? Because when we talk about the nomenclature of long-chain fatty acids, the first carbon after the carboxylate group is an alpha carbon, and that's a keto group there, so it's an alpha-keto acid. And when you think about it, when you go ahead and knock off that CO2 group, you build up a lot of negative charge. And so what the TPP does is serve as an electron sink. That's what's going on. Here's the structure of the enzyme. Here is a proposed mechanism for this. Now, again, take everything with a grain of salt. Take everything with a grain of salt. But this is one mechanism that kind of makes sense. So here comes your TPP. Now, in this case, we're minus the shrubbery. We're just looking at the business end of the molecule. And somehow that has to come off there, that proton. And we now think it's the cofactor, when it binds into the active site, gets it into such a conformation that it can pull it off. Then you have this carbanion. So here is the pyruvate here. This is going to attack the carbonyl carbon right here, because we're trying to get that carboxylate group as CO2 off. It attacks here, and then they think there's an acid that puts an OH group here. Now, the electrons get fed into here, that gets fed into here. You can start moving electrons along here. But the bottom line is that bond breaks, and there's a buildup of negative charge, but now we have the CO2. So after the lecture, go and look at these slides and see, make sure that I drew the arrows correctly. But you're building and pulling them off here, and then there's resonance contributor, which helps because you can pull that off here and have a carbanion here. All right, so this is resonance forms going on here, and in this case, you've got a carbanion. Then they think there's some active site base that's going to pull that proton off here. Those electrons are going to go in here. You've got a hydrogen coming in here, or a proton, sorry, that's going to bond to this carbanion. This is going to collapse, and now you have acetaldehyde. Interesting. But this reaction, you cannot do this with a regular cofactor because of the buildup of charge. So when you see TPP enzymes, they usually have a carbonyl carbon here, and some you're doing a decarboxylation reaction, and you have two groups next to each other. All right, so next time, last lecture, lecture number 15, I'm going to tell you about some other cofactors. One of them is PLP. It is a stunning cofactor. I know I use that term too much, but I shake my head sometimes thinking about how these reactions occur and in the time frame that they have to occur. I knew nothing about PLP until I started working on these enzymes that are involved in unusual sugar biosynthesis, and there are a number of them called aminotransferases. So I had to learn a lot about the PLPs. And then we're going to talk, as I said, about one of my favorite, the cofactor folate, and we'll talk a little bit about S-adenosylmethionine and coenzyme A. So I will see you next time.